So yeah, the future is here. So actually, from my point of view, the future took quite a long time to get here. I started studying artificial intelligence at Sussex University back in 1989. So it's great to see where we are now. And I think, yeah, there's lots of great things happening, and it's an exciting time to be working in this area. Just a theme that was talked about earlier, for making AI work, you really do have to have a good understanding of the domain, the specific area that you're working in. And I'm going to be talking today about drug discovery as the area that we're applying smart content and AI to. So in that sense, there's a great ambition here to use AI to cure diseases, to come up with new drugs. And so that's, in a sense, this idea of a moonshot, is having your high ambitions, doing great things with this technology and with content. And you may have heard of the Obama-Biden moonshot to cure cancer. And that's a really nice ambition, but I really like this quote from Harvard, which I think puts it in context that getting to the moon is like one destination, but a disease, and specifically cancer, is actually multiple different destinations. Many diseases that we talk about are actually multiple different conditions, and to actually create cures, you need to understand the individual makeup, the human phenotypes associated with those. So it's a real challenge, and it's one which people have a lot of hope around artificial intelligence delivering results. And thus far, that is still to be determined. We're still understanding whether this great opportunity in the health space and the drug space is actually going to bear fruit. But I like the ambition. John Baldoni, I was on a panel with him a few weeks back. I think his ambition is great, and he's doing some great things at GSK. This little diagram here shows you the challenges associated with developing drugs. It takes years and billions of dollars just to get one drug to market. Now, at Elsevier, we do some pro bono work. We work with a charity called Find a Cure, and we've done some work to try to identify drugs to be repurposed. In this case, congenital hyperinsulinism, which this picture really brings it home to you. What we're trying to do here is help cure diseases that our family and children could be suffering from. So it's a great use of AI technology. We focus in drug repurposing, which is a good place to start because you can get cures to the market very quickly because those drugs have already had their safety analysis done. So I'm going to talk today about smart content. So Elsevier does a lot of work in curating content and then using that to take the next step beyond the known assertions and facts and statements that you can find in the literature to actually predictive analytics, so moving beyond that information to infer new insights based on the patterns that we can see in various data. So again, I'll talk about the purpose. The purpose, because always with AI, you really need to have a domain that you're focusing on to give you the context for which you're going to use this technology. So I presented, launched Datathon Challenge back in October in Boston at the conference of the Pastoria Alliance, which is a pre-competitive group of pharma companies. So they're working together to try and develop best practice in how to apply AI to drug discovery. And through Find a Cure, which is a UK-based charity, they put me in contact with an American charity, Cures Within Reach, and their partner organization, Mission Cure. And we are making our platform available to a host of pharma, data scientists and consultants and technology companies, including competitors of ours, who are taking part in this Datathon to try to address a condition called chronic pancreatitis, which is a horrible condition, the hereditary form of which afflicts children, and one of the key phenomena associated with that is intense pain. So it's a very nasty condition. We're working to try to identify drugs that can be repurposed to treat that. And as I say, we're using our Intellect Platform to deliver that. So this is the platform that Elsevier has developed for our own data scientists, and we're making that available to be able to perform AI on the data that we've curated. So I'm going to talk about that today and try to put that in context and give you some illustrations of that. So there's really 3 pillars for this, and you can think about this the best way to apply AI. The first is to ingest information, so that's to bring unstructured and structured data together, to integrate it, aggregating data to give you your resources, your content resources in which to work. The second pillar is connection. So you're looking to create a semantic graph of information so that you can navigate that and identify the insights which you're looking for. And that's, if you like, a traditional content innovation. But going beyond that to predictive analytics is where you then apply machine learning, is mostly what we're talking about here, to then move beyond what's known, looking at the patterns and infer new insights. So we've got a lot of different pipelines in terms of applying technologies, as you can imagine, to curating information. And we have a lot of products in the life science space, and what we're doing is we're bringing that data together into the platform so that it can be accessed by data scientists. So one of the pipelines we have is used for adverse drug reactions, which is a challenge, as you can imagine. As the aging population grows, many people are having to take multiple different drugs, and so drug interactions and adverse drug reactions are really important to address. So we use machine learning at both ends, at the ingest end and also at the other end to do the analysis. So you bring the data in, you identify concepts, you map them against your dictionaries, and then you look for relationships. And that gives you the logical information that you can do inferences with. This is a sort of, you always have to have a little flow diagram when you're talking about this sort of thing, so here we are. What that does is it gives you very rich semantic data. So it's the relationships, the entities, and in this case, it's processing adverse drug reactions. But it's the kind of technology pipeline that you can apply to any form of concept if you write the right dictionaries and ontologies. So we bring that in. The second pillar is connecting all of that together, and I'll just talk a little bit about what we did with Find a Cure in terms of using the semantic data. So you start off with understanding the biological processes in which a disease takes place. This gives you a network graph of the interactions of proteins in the body and the things which they do. Essentially, what that allows you to do, in this field, they talk about targets. So it's genes or proteins that you are trying to interact with and act upon with your drug. So you start with your disease model. You then say, okay, what are all the targets associated with that? And then you say, what compounds, chemicals act on that target to intercede in that biological process? So you get a whole bunch of data, as we say, big data. And then what we do, we have a couple of databases that this is drawn from. The Reaxis medicinal chemistry is where we have the bioactivity data. So it basically says, okay, of all these potential drugs, which one is the most active, the bioactivity against these targets within that disease? And then we rank those based on their bioactivity, and we also filter them based on how many targets they address. Because if they go to too many targets, they're not a great drug because it just interacts with everything in the body. The other piece around this is validation. So one of my great concerns is validating neural network models. Which ones are really working? The trained neural networks that are actually going to deliver results for you. Because, you know, we talk about bias, but also reliability, this black box. You have to try and get to the point where you can judge these models, and that's really critical. So one of the things we did here was using our data, it enables us to then judge the models, because we can say, okay, well, sirolimus is actually a drug that does treat this condition. Not necessarily the best one, but it's one that treats the condition. So there's a sense of validation there. But essentially, you know, that's part of the journey, creating the semantic data that you can navigate, but that is knowledge that's already there. It's latent knowledge which you can explore and find. But what we're talking about now is serving that up for AI. So as I said, we have this Datathon competition, which is running right now as we speak. There's loads of data scientists taking part to try and address this problem with the data that we've provided. We've This is a cartoon picture of what we've given them. There's targets, so there's the proteins that you interact with, the diseases, and the implications. So it's saying, what do we know are the targets associated with this disease? And then we have the substances that interact with those targets and their bioactivity, and a whole bunch of associated data with that. And then what you can do, the really interesting thing, is then you can start to build these things called fingerprints, features. So you create an information space. So in this case, you start with a chemical information space. There's different representations of chemicals, Inchi codes, smile strings. You can build that into a vector, which you can then have as a data point for your neural network. And then on the other side, you can do the same with proteins. Each protein has a sequence, and you can do things like look for similarities between proteins. So this one protein might be known to be involved in this disease. Maybe you don't have a great knowledge of the disease. Maybe there are other proteins that actually are better targets. So this is the sort of thing that you can identify. And this model here is basically saying we're going to build an information space around the chemicals and around the proteins. You run that in a kernel-based machine learning model, and you can predict bioactivity. This is the idea. You can go beyond the known information, off-piste if you like, and find the patterns that could give you clues about new knowledge. And that's really the challenge here with AI, is using AI on the data and the content to infer new insights that are not actually already in the published literature and the databases. So as I say, trust in your answers. For me, that's really the critical thing, is building these models, but also validating them. And in future, certainly in the area of life sciences, it'll be a question of auditing that. You know, if you develop a drug based on a machine learning prediction, you better be damn sure that you've captured that. If there's an audit to come and say, well, what do you mean you've lost the training data? You have to be able to say, well, this is very reliable, and we've captured this. And the flip side is that you can potentially have a whole range of different neural networks or different machine learning models that are trained up. And you can look at your library of trained neural networks and do things like transfer learning and judge which is the best model for which use case. So that's all in the future. Well, no, it's all now. That's what we're doing. So just to wrap up, as I say, we're running this challenge at the moment. This is a Slack channel for the data scientists out there that potentially use this. So we've got a whole bunch of people interacting, communicating on this Slack channel, building different models, running them on their Jupyter notebooks, running Python code and doing great things with this data. And we'll also feed that back to the Pistoia Alliance because we're looking at best practice in AI in the life science space. And with the following wind, we might also find new, repurposed drugs that can treat this horrible condition. So the winners, if there's such a thing saying, from this Datathon competition will be announced at the Pistoia Alliance Spring Conference on 13th of March next year in London. And watch the space. I'm hopeful that we'll get some great outcomes from that. And then, if that turns out to be the case, Cures Within Reach and Mission Cure will take that forward to a clinical trial and hopefully out to the patients. So, thank you very much.
Brilliant, thank you. We've got time for some questions. Does anybody have a question? There's a mic coming towards you.
Hi, that was a really fascinating presentation. Thank you. I was just wondering if you go back to the obligatory flowchart, because I just wanted to see if someone would take a photo and then put it up on Twitter so I can look at it later. So that was the request. The question is actually about the technologies you're using to stitch this series of models together, this process. What are you using, if you don't mind, if you're able to share, some insight into your stack?
Gosh, okay. Well, all of the information is stored on an ADFS server. But as I say, we're pretty agnostic in terms of the tools, that are used to support this. So there's TensorFlow. There's a whole range of different technologies. So the whole purpose of having this platform is to enable the data in a open format to come into the platform, and then you can run, you can run R, you can run Python, anything you like, basically. And one thing I didn't mention, we're very interested in FAIR principles, which, for people who are not aware of this, it's a protocol for data sharing that allows you to then bring information in from all kinds of different sources. So our approach is to be as open as possible and to be as flexible as possible. Yeah.
One last question, yes?
Hi, Dinesh.
Thank you very much. Great presentation. 2 questions. Is the sources restricted only to Elsevier? Or is the source of content restricted only to Elsevier content, or is it?
No. So the databases are Pathway Studio, which was a database we acquired a few years back. So the science is curated from all of the journals, so it's not limited to Elsevier.
And so then how is it different from GeneGo or Ingenuity?
Well, they're great products. I wouldn't disparage them.
Same as this.
It's similar. Essentially, what you're talking about is curating the data, getting the semantic data in place. What we're trying to enable here is an environment that allows the data scientists to then take that semantic data and very straightforwardly create features for it to put into their machine learning models. So it's an enabling platform, essentially.
Okay.
Yeah.
And it's a subscription model.
Yeah.
Thanks.
Brilliant, thank you. Can we have another round of applause, please?